Hi, this is Dr. Ben Morrill. Welcome to episode 51 of Reptile Genetics Weekly. Glad to have you here. And we've got good stuff to talk about today, once again. So this week we had fast tests going out last night and today. We got more going out this week, at least one or two more times. So lots of fast tests. Thank you all for using our services, and so thankful to be able to get those to you quicker now. The big run, so for people doing full panels or doing the few mutations we don't have a fast test for yet, that run I'm actually starting today. Actually, while we're recording this is Wednesday. I might have a package come that will give me all the stuff that I need to get started on the big run today. So over the next few days, I'll be prepping that. And next week, probably like Thursday next week, so about a week from now, I think that's like April 11th. That's probably when the big run will have all those results back for full panels and tests that aren't fast tests yet. But like we've been saying, by the end of April, we should have everything switched over to being a fast test. So then it'll just be the full panels that will take a little longer. But speaking of that, we've been talking, now that we're going to have fast tests available by the end of April for all 30 mutations that we have tests for right now. We had a question for you all, and we'll actually have it available as a poll. But feel free, if you have more thoughts, feel free to let us know in the comments what you think. So the question we have is, if we had all three of these available, what would you use the most? Because we might not be able to make all of these be available, but just have that be the stipulation if these were all available. So if you had a choice of 22 gene panel, and then over the next couple of months switch that to a 30 gene panel, where everything is a fast test. So you could say, hey, I want a fast test done, and we would run all of them as fast tests. It will cost more, but you would have those results in 1 to 2 day turnaround. Let us know, hey, if you had that available, I would definitely use that. Or would you rather have the second one where we say the custom pick 5, and maybe we would make an option, hey, I want to do a pick 5 or a pick 10, and so it's completely custom. We'd have two different prices, a pick 5 and a price and a pick 10 price, but then that's completely up to you. But all of those would be run as fast tests. So if you're mostly interested in 5 or 10, you could get that done. Or would you still want to do that full panel? So that's kind of what we're wondering there. If you have the option to do a pick 5 and a pick 10, would you do that more than a fast panel, or a normal panel that's a lower price but takes longer? But then the third choice we have there is the genomic registry. So with the genomic registry, you would send a shed in one time. We would sequence the whole genome and keep that information. And so later on, if you wanted any other test done, say when we get Monarch or Stranger or any of these others done, that you want to know the results for that, you wouldn't have to send in another shed. You just have to let us know, hey, I want to update my genomic registry results for this animal, and any new test, whether it's 1 or 2 or 10 new tests that we have, it would automatically update your animal for everything. There would be some small fee for that to rerun your data, but yeah, so the genomic registry would take longer. But it would make it so you don't have to send something. It also could be something, if you have a multiple paternity clutch, you could send in sheds from the babies, and we would be able to tell you, which babies got sired by which male. We would need the sheds from the babies, but you wouldn't have to send in anything from that animal that was already in the genomic registry. You could also send in a shed from an animal and say, hey, this is supposed to be related to, the very first, the very first VPI xanthic desert ghost clown. Is it really? And, if that's already in our database, we'd be able to say, yes, it is, or no, it's not. So there's a lot of different things we will be able to do with that registry. But anyway, like I said, let us know in the comments if you have more thoughts, or at least do the poll and let us know if you what you think you would use the most if all three of these were available. and that will help us to know what we try to work towards the most to make available for you, so you have that choice. So once everything we can do is a fast test, then we'll have some more options that we'll be able to offer to you as customers. So but yeah, with that, Kayla, how are you doing?
Hey, Ben, I'm doing great. Just playing off of the last thing you talked about with that poll, if it should already be live by the time that you're watching this video. So at the end of the video, after you like, comment, subscribe, do all of that stuff for us, go to our YouTube, go to our main YouTube channel. So right below the video, this side actually, give it a click and it'll bring you to our main page. Hit the community tab, and that's where the poll will be. So just making sure everybody knows in case you don't see it already.
Yeah, well, I was telling Kayla as we started this, like, somebody like me, a lot of you that are younger, you probably know how this all works. But I was telling Kayla, any of them that are older like me, they're not going to know how to do this. So
we got you.
You said we might be able to have a link in the description as well.
Possibly. I didn't find a way to share it. So
you might have to go.
Might have to just go, but I mean, it's 2 clicks away from here, from the video, so it's not hard. Don't be scared.
So for those of you out there that are like me and have never been on the community tab, this is the time to give it a try.
Yes. It's not scary, I promise. But I have really good news. So snake update. So, you guys know how my first clutch ever was a really rough hognose really rough hognose clutch, which is still in progress. But just in time for Easter, I woke up to a beautiful, perfect clutch of rat snake eggs. Awesome. And there's a picture of Mama Chickadee. She is a het lightning western black rat snake, a little pretty locality morph that was found in, well, you'll see what the actual morph is in a second here. That's on the right. That's the sire, Mr. Lemon Bars, who proved out beautifully. No slugs at all. Perfect first clutch.
It was an even dozen, right?
Even dozen, exactly. Which is a really solid first clutch. And she did great. I actually didn't get any pictures of it, but I tried something out that I heard someone suggest where you like put a much larger lay box for her and fill it with like peat like cocoa coir and sphagnum moss. And I gave her like a bunch of substrate that she could bury it in, and because I wanted to replicate wild nesting behaviors. And sure enough, she laid in there and she like laid each egg and covered each of them completely with bedding, and then packed them all down. Yeah, she packed them all down really, really tightly, to where I actually had to like excavate the eggs, and like she had, like, it was actually a hard packed substrate. Yeah. Yeah, and I mean that's what they would do in the wild. They'd find some sort of like composting pile of composting, usually nesting material of like plants or something like that, and the decomposition kind of keeps the eggs warm even if there's like a cold spike or something. So got to replicate some wild nesting behaviors in her, and it was great.
Cool.
Yeah, so really excited about that. Hoping for a nice 50-50 clutch. So hopefully half and half wild type, half and half lightning hypo. So awesome. Excited.
Yeah.
Very pretty babies. My hognose steak is still fat and sassy. She dropped 2 more slugs for me yesterday, so 3 more left. She's looking great. A little more deflated now. But still ferocious. So we're doing good over on my end.
Good.
Yeah.
That will be such a good day when all of those are out for her.
I know. I'm grateful to Tom Harbin and Jeff Galewood for helping me out with that. And saying that no, you probably don't need surgery. Sometimes the girls just take 4 months or so to lay their eggs. So not surgery, but just mild panic attack. It's fine. Anyway, so tell us about what tests we have going on. What's new with our genetic testing, Ben?
So with the fast tests, those of you that saw this last week, nothing is new yet. So we're April 3rd. So we're just
Yeah.
starting in April. So we still have the same ones available as fast tests. Next week, we'll likely be able to add the ones here and maybe some more too.
Woo!
But yeah, we're
That'll be
still
cool.
on track to make it by the end of the month. So we'll have all 30 ball python morph tests we have available now. All 30 should be fast tests.
That'll be great.
Also, sex determination for ball pythons we'll likely be able to get. But with running on our side, those of you that are interested in sex determination for ball pythons or colubrids, even though they're not fast tests yet, we have been able to run them more quickly. So it's about a 1 week turnaround now from when we get your shed to when you get sex determination results. So it's better than it was. Sometimes we weren't able to run it for 2 or 3 or 4 weeks, but that's getting faster now.
Excellent. Glad to hear. Let's see. And as for the tests that are currently in development, I know we did a research run a couple weeks ago. So what have been sticking here so far?
Yeah. So thankfully, we'll have a whole bunch more information on the first 4 there: Monsoon, Monarch, TSK, Xanthic, and GCRA Xanthic. I know many of you have been waiting for.
Oh, yeah.
So we'll have even more information on those. We'll have data for Barnhart Black Pastel. So we'll have a chance to have an actual specific test for Barnhart Black Pastel. And Paint Sentinel, we got a lot of sheds from you all, and then also Acid and Confusion. Thank you all so much. We got lots and lots from several people. So we'll have a very good chance at those tests. Also lace, white lace, we have a shot at, and then there's several others that we also have a better chance. So this over the next, I'm hoping by next week when we do this show, I'll be able to give you an idea, especially 2 weeks from today. I'll definitely have an idea. I won't be able to know for sure what tests we have, but I'll be able to tell you which ones look like we have some good hits for. So
yeah, definitely. I know white lace or lace, we were looking especially for more sheds. People came out and did a really great job sending in the Paint Sentinel and Acid Confusion sheds. We got oodles of those. So y'all did good.
yeah,
Lace could use a few more. But
definitely, especially white lace, the homozygous
yes.
one. We can get a few more of those. That would help a lot.
And for those of you wondering about the pictures on the page, left side is a white lace clown by Mutation Station, and the right side is a, correct me if I'm wrong, at least the top, the orange baby is it's from Tom Harbin. It's a Monarch G-stripe clown, I think.
Yep.
And then the other one's just same thing minus the Monarch. So
yeah, pretty cool animals.
They were cool, so we wanted to put them up here as visuals. So yeah. So it's time for a monthly Morph Spotlight, which we've been doing more than monthly, but that's okay because you get to learn more.
We got a lot to go through, so we'll be more like every other week for a little while.
Yeah.
So yeah, this time we're going to cover the last one in Dr. Hannah Seidel and her students, the paper that they put out where they looked at albino, ultramel, and lavender albino. So lavender albino is was published. The work was done by them, published by them, and a test that she even made available and everything. So they did a lot of really good work in that first paper. And so thank you once again for Dr. Seidel and her students at Eastern Michigan University.
Yeah.
But yeah, so with lavender albino, it's like we talked about the last morph update. We were talking about ultramel where there's 2 different mutations. With lavender albino, so far to date, there's still only 1 mutation. And it's actually an interesting one. Of the mutations so far, it's a pretty large deletion. It's 1,514 base pairs, so 1514. So 1,500 base pairs that got cut out. And so in the paper, they actually show the gene. So the ACA2 gene, OCA2 gene, that's a gene that's very frequently, if there's an individual that's albino, of a different species, whether it's ball pythons, corn snakes, people, mice. OCA2 or ACA2 is very frequently the gene that gets interrupted or changed. And so if you look in this figure in the kind of pink or light reddish, you can see, well, I guess, first of all, the ACA2 in the top left, those parts that are black blocks, those are all the sequence for the OCA2 gene that's actually part of making the protein. They're called exons, I mean. And then the lines are all introns. So that's DNA sequence that's there, but it doesn't do anything specifically for the ACA2 gene most of the time. So anyway, those blocks, you can see all of those blocks spread out. And the one where the pink deletion, it says deletion for Lavender albino, that 1,514, you can see that completely takes out one of those exons. So that chunk of DNA that's important to make the ACA2 gene work, that gives you melanin, that specific exon gets completely taken out, and that makes it so you no longer get melanin, and you get the lavender in a ball python. You get the lavender albino ball python.
Interesting. The thing I have a question about that, this may or may not relate to this paper, but from that pink bar spread across there that says 1,514 BP or base pairs, it cuts out the, did you say the exons?
Yeah, well, it cuts out that one. So right above it, there's a black rectangle. So that's denoting a chunk of sequence that is exon sequence. So it's part of making the protein that matters. And so that little bit of genetic information that makes up that black box, which probably, a few, that 1,500, you can see how wide that is. So it's probably only 300 or 400 base pairs in that 1,500 that matters.
Yeah.
But it wipes out some of the sequence before it and after it, along with that whole exon is just gone.
Okay. So the introns are the stuff that doesn't create proteins that doesn't code for anything. Is that what people refer to as like junk DNA? Or
exactly, that's what we used to call junk DNA. Yeah. So when I was in undergrad, I took a biochem class, and that we just assumed that was junk DNA. It doesn't do anything, but we know now, after getting the whole genome, there are times where it definitely makes a difference in gene expression, and there's something called alternative splicing, where you can make, even with those black blocks for ACA2, there could be an alternative way where it uses the first 7 and the last 2, and it makes a different protein. The information is there for that whole OCA2, but if you do some alternative splicing, you might get another protein from that same genetic information. And so some of that, like how, to tell the genetic machinery when to do that alternative splice, some of that information is sometimes in the intronic sequence, or there's like recognition sequences that will cause cuts, or there's many different things that now we know what was junk DNA actually does do something sometimes.
Interesting. Okay. Well, that might be a fun conversation for its own episode in the future, maybe.
Yeah. But on the far right, where there's that long intron, so you can see the last 2 black rectangles, there's that long intron. If you took 1,500 base pairs out of there, then you probably would still get a normal looking ball python. But the fact that it took the 1,500 base pairs right where there was an exon, then that messes up that protein. It doesn't function properly. You don't get melanin, and in the end, you get a lavender albino ball python.
That's so interesting. Okay, cool. Thank you for explaining. Let's see. And so, like we said, it was mapped by Dr. Seidel, like with the last few episodes that we did for Ultramel, A and B, and a few others. These are the same group of people who donated or contributed sheds for mapping this gene. Again, lots of familiar faces in there.
Yes.
And so, Ben, how do you grade this with our special RGI grading scale?
Yeah, so since it worked very well in Dr. Seidel's hands and her students. With us, other people, it's been published in the scientific paper. It's definitely been, run over 1,000, several thousand times by now.
Yeah.
And many of those samples were known, either lavender albinos or hets, and it's always matched up. So it's an A plus. It's a very, very good test. Dr. Seidel and her co-authors and students, they did an excellent job with this work, and we're very grateful for it.
Awesome. Well, sounds good. So for a shed donor shout out, who do we have this week?
So we have Hare Halla Farm. So this is Jessica Hare, and we're very thankful. I wanted to show this time.
Oh, yeah.
This is the stack that we got from her just for corn snakes.
Woo! Oh my gosh, that's all corn snakes?
These are all corn snakes.
Nice.
So she's got them all labeled. She did a great job. We know what everything is. And so that this year we're hoping to get to at least 2 or 3 other species besides ball pythons, and corn snakes are towards the top of the list because of people like Justin send in a whole bunch that gives us a foundation to work from. She also sent in some boa sheds, some ball pythons, some rat snakes. I think there was a hognose in there. So
yeah,
she really, really great job of collecting. I'm sure that took a while. She collected and notated what everything was and got it to us. And so the only way we can do new tests is people like Jessica and many of you. But once again, thank you so much for sending sheds in, and we will definitely put them to work. Sometimes like these, she sent these a while ago. We haven't used them yet, but like I said, 2024, those of you that sent in a bunch of hognose and corn snake and boa and blood python, I hope to get to at least 2 or 3 of those this year.
Yeah, I'd love that. I know I mean, personally, as a colubrid breeder, I'm excited for it, but I mean, in general, it's exciting to see this project move into different species with the morph testing. So love to hear it.
And then the other thing is, you can see there some of the animals she has available. She has a lot, with all these corn snake sheds, she's got a lot of variation. So if you're interested in corn snakes, definitely check out her morph market.
Yeah.
And she also does I don't know if it's weekly or maybe it's even more often than that, but she does do a podcast and YouTube show.
Oh, really?
Yes. Yeah. So definitely check that out. If you look up Hare Halla Farm, you'll be able to find that. It might be under a different name. Yeah, it's the Holdback Rack, I think.
The Holdback Rack. Yeah, I've heard of that
podcast. Yes. So
all right.
He has another I think it's Jennifer, but yeah, she has another host on there. And they also do auctions, help benefit breeders that, have some animals they need to get sold. I got lots of people that get on there and bid. So they do a lot of really great things to help people in the community.
I love it. Okay. And you can be a future shed donor too, for our shed donor shout out if you'd like. If you've sent us something or if you plan on sending something, let me know. Message me at raregeneticsocial@gmail.com. Give us your name, your logo, pictures you like, your info. That way we can shout you out on the show too.
Yes.
Yeah. And I think that's pretty much all we have for today, Ben.
All right. Well, this being episode 51, and our original goal was to have 52 episodes. We were just talking. I think I'm going to see if I can talk my wife into being on next week's episode 52. We'll talk to her a little bit give Kayla a chance to ask some questions. And I guess that'd be another thing you can put in the comments. If there's any questions you want to ask my wife, what it's like to live with a snake dork that's also a DNA dork.
Oh yeah. I'd love to hear some of Sarah's insight here. So also she's really fun. So I think it'll be a fun episode.
Yeah. I think the first time she ever actually went to my bedroom, I think I had like a 4 foot by 4 foot by 2 foot cage with a pair of boas actively breeding while we were in there. So At least she was warned right away, you know.
Exactly. Yeah. Oh yeah. Yeah. No, I've had similar experience. Luckily, the guys I brought home while I've had snakes have been totally cool with it. Like Jay, he's like the girl that laid eggs earlier. Like that is whenever I pull her out, Jay says, "Oh, there's my daughter," and holds her. And so that's his daughter. I've been calling him Grandpa because we have eggs now. So life is good. It's interesting. So I'd love to hear Sarah's insight on all that too. So
Sounds good. Well, I'll see if I can talk her into it.
sounds good. All right. Thank you guys. As always, make sure you like, subscribe, comment, make the algorithm like us so we can reach out to, so we can, get in touch with more snake nerds and genetic nerds. And yeah, I guess we'll call that a wrap.
Thanks.
Thanks, guys. Later. Outro in 3, 2, 1.